I'm Tegan. I'm the lead editor of Geroscience, and today I'll be talking to James Peyer of Apollo VC. Apollo is a venture capital firm focused on life extension technology. Also, James is the co-founder of Geroscience. So, James, you have a long history in the longevity space. Can you talk about how you got started?
Sure. So I I've been interested in aging since I was about 15, 16 years old. And I decided quite early on that that's what I was going to spend my life doing. Um, my initial kind of step into the space was a fascination with telomeres, because in the 90s, we were just kind of starting to untangle what telomeres and telomerase were doing in the cell. And there were a lot of theories floating around that if you turned on telomerase, you could just make cells live forever, and by extension, you could make people live forever. And so at first I dug into that, and then as I was exploring telomerase, I started finding out that almost all cancers express telomerase, and that if you overexpress telomerase in people, it probably removes one of the checkpoints for cancer. And that seemed like a problem. So I was like, well, maybe we have to keep telomeres long, but actually get rid of telomerase. And so it was through that question, or that kind of line of reasoning, that I ended up meeting a gentleman named Aubrey de Grey on a forum, a web forum in the early 2000s, and started talking with him about his interesting ideas about telomerase. And then as a young man, I kind of like just grew into the rest of what I now think of as the geroscience space. So yeah, I've been interested in science for a really, really long time. One of the key parts, I think, of very advanced therapies to treat aging will be gene therapies. So I decided to spend my undergraduate and graduate work on blood-forming stem cells, which are some of the early targets for gene therapies for a number of different reasons that we can talk about later. And then ultimately, I was really interested in translation therapies, which is why I went after my PhD to work for a consulting firm that helped advise pharmaceutical companies on their R&D pipelines. And then now I run Apollo Ventures, and I'm finally, in some ways, for the first time, professionally involved in aging research or geroscience proper, where we help start or fund new companies with scientists that are studying the biology of aging.
So, you were in stem cell bio, and then you switched to the business side of things. Can you talk about why you chose that path in particular?
Sure. One of the ways of putting this is that I just wasn't that great of a scientist. Like, I love science and I love thinking about science, but I looked around me at my peers, and so many of them were so excellent at asking basic research questions and doing experiments in mice and doing very technically and intellectually challenging assays. And I had seen in my lab these excellent cadre of scientists bump up against a wall when they were trying to think about their projects moving translationally. And especially, I felt like many of them, because I work with a number of MD PhDs as well, many of them would be thinking about the aspects of their work in patient care, but they would miss a little bit of a gap in the middle. And that gap was primarily driven by economics, chemistry, which not all, like as a geneticist, like not a lot of us spent a lot of time thinking about chemistry, and then like business and business relationships. And so I had started my first company when I was 21, started a small hedge fund while I was a graduate student, and like so these sorts of things were just kind of part of my personality already, and I slowly began to realize that something that I could offer the community was trying to build a bridge between these excellent basic scientists and the broader business world that eventually leads to patients, but it felt like there was a bit of a gap there. And certainly there are lots of other people besides me who are occupying that space, but certainly in the fields of stem cell biology and now aging biology, there haven't been a ton of people really specialized in that space.
Can you go into more of the specifics of this gap?
Sure. So there's a commonly used phrase in the field called the biotech valley of death, which is like right at about the point where you've got a really interesting assay or a validation of an animal model. You've been able to attract grant funding, and you have your scientists of whatever flavor, professors, PhDs, postdoctoral students, pushing that science forward. And then it reaches a point where it has to spin out into a company and attract some new early stage funding. But that funding is now so far from the clinic, right? You're still 9 years or so on average from your market approval for that drug, that finding the funding to go in a serious way to build that technology into a therapeutic that will make it all the way through your preclinical development, clinical trials, and then market approval is very rare. There are a few groups around who do that very, very, very well, especially like Boston, San Francisco, London-based biotech venture capital firms. And then pharma companies can do that very well. But there aren't so many of those actors compared to the quantity of research being generated in every major city around the world. And so a lot of biotechs get started and then almost immediately stumble by failing to find funding or finding the right strategy to go forward, which is why we call it the biotech valley of death, because it's a tough transition where someone who's a basic research scientist enters a world where all of a sudden the next step for their invention has to do with all of these fields that they've never really had to care about before. And so there's quite a transition that needs to happen. And there are a couple of those in the biotech world.
So in simple terms, what is the main bottleneck? Is it primarily funding?
I would argue not really. It's a combination of money and strategy. I mentioned just now that there are a couple of transitions that let's say a compound has to go through in its development, right? When it goes from understanding the basic science and kind of the development of an assay to the development of a compound, and then usually the optimization and toxicity testing for that compound, which is a completely different field, right? It can go from basic biology to animal models to like synthetic chemistry and physiology for the tox piece. And then it switches again and goes to the clinical piece where you need to say like, okay, here's how we're going to appeal to clinicians and so on. And then it has to switch again to go to the market piece where you talk about pricing and reimbursement from insurance companies and so on. And each of these steps, a lot of groups have a ton of trouble crossing from one side to the other. And so finding a partner that can enable a company, or let's even not call it a company, let's call it a project, right? Enable a project to jump from A to B to C is really the biggest value there. And yes, it does take a lot of money, but I think money is just one part of the equation.
Is the regulatory structure a major hurdle with translation?
I don't spend too much of my time talking with the FDA or the EMEA in Europe. I think that they have pretty clear rules, and especially in the last decade have been much more responsive than they ever have been in the past. I think that overall the regulators are doing a pretty good job of things. The tougher part in my mind has been the stuff that comes before the FDA. How are you identifying and validating your drug before you ever start a clinical trial? How are you optimizing it to make sure that it has the maximum chance of being safe in humans? How are you setting up your company so that even if the first drug that you go in fails for some unforeseen reason, that the whole project isn't jeopardized from this one unexpected failure? These are all the sort of things that I think are the biggest stumbling blocks for early stage biotech companies that I spend most of my time dealing with. Certainly regulatory approval is a big one that we're always thinking about, but the whole company should be built in such a way that regulatory approval is achievable if you meet certain clinical milestones, and the return on investment for investors is certainly there in pharma. We can talk a bit about that later, but from a returns perspective, it's actually has been in the past decade better for investors to put money into early stage biotech companies than to early stage tech companies. Which, given how everyone talks about Facebook and Airbnb and Uber and all of these other things, it's kind of an interesting statistic that pharma companies have actually been doing better.
Yeah.
But yeah, so even given that, the point is that the return on investment is there, the regulatory structure is working and rewarding those drugs that succeed. It's just that not all of them are able to get there. And that's partially a funding bottleneck and partially a strategic bottleneck.
As an investor, how do you choose projects with good prospects?
So if you ask any VC investor, they will give you some statistics about what they call their deal flow ratio, which is basically how many projects do I see every year versus how many do I invest in. And the reality is, for almost all investors, you see a whole lot for every one that becomes an investment. Apollo is a little bit different than many other investors in both our appetite for risk and stage, but also our willingness to go in and build something that doesn't exist yet. So we have a very clear focus in the biology of aging, right? Which to us means a rather defined set of things. And so we have been going out, seeing scientific presentations, often even before there's a paper published, talking with researchers, going to conferences, etc. And then because my whole team, my diligence team is scientists, we go in, we get excited about some specific scientific aspect. We've all been trained a little bit on the translational side, like seeing what other biotech companies are doing, and then jointly hatch plans to say like, okay, this is how this would work if we turn this into a biotech company. And I think that starts with just exceptional science that is right at this unique stage where it's ready to jump from bench to biotech, let's say, right? Where it's an academic project that has proof of concept in some way. So sometimes in animal or even in animal models, whether it's mammalian or a lower organism, and then potentially a compound that already exists in addition to a target, but usually not an optimized compound. And then we jump from, and if it hits that check mark in a way, where you've got this, it's a very, very cool, novel mechanism of action, then you go and dive one level deeper to say like, okay, is there a way to turn this into a biotech company that makes strategic sense? And if all of those checkboxes are hitting, then you say like, okay, how can we make this work? And then you have to go through the next steps, which is pulling together a team, launching something and then growing it. But I think that how we find it starts and ends with identify great science in the aging space.
Is there anything in particular that you avoid, anything that you watch for as a bad sign?
Interesting. Yes, tougher question, but I think a good one. So red flags to me as an investor include going for dementias as a primary indication. There's all sorts of really, really fascinating research going on right now with Alzheimer's disease. And I think that there are a number of interesting theories, including ones about metabolism, about lysosomal degradation, different ways of targeting both tau and beta together, et cetera. Like there's all sorts of interesting ways to go after Alzheimer's disease, and many of them apply to Parkinson's as well, that are fascinating. And I think some of them are absolutely going to work, but the history of biotech companies makes investments in this space in specific projects that only have applicability to Alzheimer's or Parkinson's, or want to go after those things first, incredibly difficult to fund. Alzheimer's trials, there have been 200 or so since 2000, and there have only been 2 successes out of those 200, and neither of those has created a disease-modifying drug. And so while there's still a lot of appetite for something new, being able to wrap those new things in a shell of like, there's a safer piece to it and then a more exciting piece behind it is kind of core to how we think about the Alzheimer's space from a business perspective, even if there's from those parts that I just discussed before, animal models and a market strategy and good scientists and so on, even if those are all check marks for Alzheimer's, we're still always a little bit cagey about it, just because it takes so much money and it's so risky to do a big phase 3 Alzheimer's trial. I would add to that especially in our space, right, in this Geroscience space, most of the work suggests that intervening early in Alzheimer's is the best way to intervene and prevent the continued degradation of memory. And that makes the trials that you would potentially perform for a drug like that even longer, tougher, more challenging, with a higher bar for safety and efficacy.
You mentioned primary indications. What are those? Can you talk about those?
Sure. So when you start a biotech company that's focused on making therapeutics, you want to have a drug that's approved for an indication, right? You're going to be going and after you complete your preclinical development, which is basically you test whether the drug works in mice, and that it's not too toxic, and that it's not broken down in the plasma the moment you take the pill. Then you have to say like, okay, now we're going to start a clinical trial, and that clinical trial usually has to be for a specific disease. And in the aging space, particularly, we are blessed and cursed with the fact that things in Geroscience that extend the lifespan of animals, or target one of the hallmarks of aging and are able to kind of reduce the buildup of or rejuvenate the damage of aging, can often be applied, at least in the animal models, to a ton of different diseases. For example, when we eliminate senescent cells from the body of a mouse, we see muscle regrowth, bones get stronger, rates of cancer go down, lifespan goes up 35% at maximum, hair can regrow, kidney function improves. There's all sorts of things, cardiovascular function. So when you say like, okay, well, what do you now make the primary indication you're going to design your first clinical trials around for a drug derived from this? It can be quite challenging. And so just to use a very concrete example, to continue with the senescent cell space, Unity Biotech, who are putting together small molecules to kill senescent cells, are going after osteoarthritis as one of their first indications. But I think that we all realize that the senescent cell space is intended to eventually be bigger, right? Ideally, we would all be eliminating our senescent cells for all of those things at once, right? Restoring our muscles and bones and whatever else in old age. But we just can't do that in the current regulatory system right yet. Or perhaps even more accurately, we could, but the clinical trial risk of doing so doesn't make sense from an investment perspective, but also from like what exists in the insurance industry and like what's going on in doctors' offices right now. We need a little bit more time to validate the geroscience approach to get there. So that's what I mean by primary indication. And then once you have your primary indication, then you say like, okay, we're going to go for this one thing first, and then it's going to branch. And as soon as we get approval for the one thing, and this is an important part to understand, it's an important part to understand that because most clinical trials don't work out, for any given project, the biggest hurdle that you can make is once you show that your drug is safe and effective in human populations, then all of a sudden it becomes very easy to say, okay, well that biology is also relevant to disease B, C, D, E, and F in addition to A, right? And so most investors will say like, instead of advancing A through F to the clinic all at the same time and incurring the same costs throughout those things, it's worth spending the little bit of time to stagger those things, go with A first, and then plan B through F to kind of follow up as soon as there is validation that A is really going to work. And that way you could actually throw a lot more things at the wall, right? You can try 10 projects instead of one, because you're going to be doing 10 different A's for 10 different projects, as opposed to one project with 10 different indications that you go for right away.
Interesting. You said something about validating the geroscience approach. How does one validate the geroscience approach?
Well, this is not my particular topic of expertise, but this is one I spend a lot of time thinking about. So let's first define the geroscience approach, right? So the geroscience approach is the idea that by altering the fundamental biology of what makes us age and decline, we can prevent the diseases of aging, or at least delay the diseases of aging, making us healthier for longer. And sometimes that means also living longer. And I would say this includes topics such as, metabolic dysfunctions that are incorrect, such as the rapamycin metformin type studies, but also, altering our senescent cells, messing with our telomeres to make us live longer and healthier, et cetera, et cetera. There are a bunch of different approaches that have been hypothesized or already shown in animal models. By validating the geroscience hypothesis, I mean that we haven't yet shown in humans that we can take a single therapeutic agent, give it to a person, and have that person be protected from multiple diseases of aging, or cured of multiple diseases of aging. But I think that that's even a further stretch. Let's even just say preventive. Once that happens, this regulatory structure that we've been talking about, it changes substantially. Because once you can approve medicines for the prevention of multiple diseases at one time, then the game becomes very different. And you don't have to look for your one little thing that you start with and then go branching out to other things. And so this is why, and I know on Geroscience you've covered the TAME trial before. This is why I'm such a big advocate of the TAME trial and similar other trials for NAD boosters and and other types of geroprotectors in human studies, because I think that even a small success, right, like a regulatory approval that shows that metformin, which is obviously not a great life-extending medicine, but does have a serious effect, right? In mouse studies, you're talking about 5 to 10%. If that has an effect in humans, which could justify insurance companies paying for metformin for people at a high risk of developing an age-related disease, it completely changes the game for people doing senescent cell research or telomere research or stem cell research who want to rejuvenate the body. Because all of a sudden you have not just a precedent of FDA approval, but you will also get out of that biomarkers and ways of making the clinical trials go faster that you can use in future trials. And so that validation of the geroscience hypothesis, I think, will actually be a watershed moment in the history of medicine in the 21st century. Whether it's the TAME trial or NAD or any of a number of other compounds.
Circling back around to primary indications, are there any in particular that are better or worse?
So one of the things that we specialize in at Apollo, and one of the things that I find really, really, really fascinating, is that geroprotectors, or let's say therapeutics that interfere with lifespan in animal models, we usually know which molecular targets they're going after. And so let's just use a very basic example, which is rapamycin goes after the mTOR protein complex, right? Mm-hmm. And the mTOR protein complex is certainly involved in aging, because when you inhibit mTOR, and specifically mTOR1, lifespan of mice goes up. And it seems like that's probably going to be true in dogs and may even be true in people. We'll see. And rapamycin is one decent tool for that, but a number of groups, including us, are working on other tools for interfering with mTOR as well. But we don't want to start with treating all diseases at one time in a TAME trial with mTOR. And so what we look for when we think about the primary indication for these things are specific diseases that have a very high unmet need. And when that unmet need is really, really high, then we can launch the drug targeting often a rare population of patients that really have a problem there. So when we look at the mTOR space, for example, there are a number of diseases in which mTOR is chronically overactive. And this can be genetic diseases, genetic diseases, acquired conditions, but they're often like very serious conditions that have high fatality rates and very few existing treatments. And then we would think about launching a drug for those things first, testing our compound in those conditions first, and then if it works, to our earlier conversation, if it works in that little piece, if it's effective and safe in patients, then we expand it out to a broader range of people in whatever way makes sense as quickly as we can do it. So the short answer is that we don't look for a specific indication, and there isn't one that I think is like the aging stepping stone indication. If we wanted to think of those there, you'd look at like sarcopenia, osteoarthritis, some of these things that are very associated with aging decline. But I think actually the more promising route is to go after the genetic disease, rare condition, things with a very high unmet need as a first stepping stone that will depend enormously on what the science underlying each individual therapeutic is.
How are progeric diseases as primary indications?
So progerias are very interesting scientific cases. And we have actually talked with some groups that are working on treatments for progerias. Um, the first thing to mention there is that different progerias are molecularly very, very, very different from each other. And secondly, one of the issues with many genetic diseases, including progerias, is that a lot of what occurs in progerias is has already happened by the time you start the drug. So unless you can be highly rejuvenative, it's often not enough. Like, you might be able to help the situation and maybe ameliorate some of the existing decline, but usually with progerias, their problems begin so early on that unless you can come with a truly rejuvenative approach, it's not going to have the sort of impact that I would want to have in going with a patient or in treating a patient. So we've looked at we have talked to a number of researchers working on this space, and I think that there are some pieces of work that we've looked at that could truly make a difference in the lives of people with progeria. But because of the very, very severe nature of most of these progeria conditions, I think that it is a very tough one to develop really meaningful compounds for. So it's a specific and tough question that you asked, but I think that's the best answer that I can give at this stage.
Are most of the therapies that you encounter preventative and not rejuvenative?
No. Most of the therapies that we encounter have the capacity to do both, but are specifically rejuvenative while broadly preventative. So, for example, with senescent cells, let's say, as an example we can come back to, or rapamycin, but let's use senescent cells. With senescent cells, there are a number of conditions that we think the pathology is really driven by senescent cells, right? Let's take sarcopenia as an example. If you can get rid of senescent cells, and then acutely that will restore the muscles of aging mice. There is also the chance that, if you take a mouse that's already very sick and very aged, and then you get rid of its senescent cells, and to be fair, this hasn't been shown in academic mouse data completely yet, but you can't immediately just restore a nearly dead mouse to health. And this has been true for, no matter what treatment that we see in the geroprotection space. So that you can intervene late and reset somewhat or pause somewhat, but once things go very bad, then at some point there's no coming back, even if you're specifically rejuvenative on some topics. However, that same treatment can be broadly preventative in that if you interact early enough, it can prevent the mouse, in this case, from entering a downward spiral that leads to the ultimate levels of pathology that are no longer sustained by senescent cells. Does that make sense?
Yeah.
Yeah. So I think that's usually what we see. And I think that the geroscience space, at least in the early days, is going to be defined by these dual acting compounds, things that could be first used against specific pathologies, but then pulled back to be used as broad preventatives. And then as the field develops, I think that it will go in two directions. One, doubling down on specific methodologies or specific mechanisms that will then have to be used in combination with each other in order to make a broad effect on the disease. And then simultaneously, there may also be specific things that are not rejuvenative in and of themselves, but once our geroscience hypothesis is proven in the clinical setting, then those compounds could be the next generation of things following your metformin or rapamycin analogs or whatever it is, that can be used to prevent diseases, but they're specifically designed to prevent diseases, not to go after existing indications.
So your academic career focused on stem cell biology. Can you talk about where you see stem cell therapies going?
Sure. So I think that stem cell biology has two roles to play, one outside of geroscience and one within geroscience. Outside of geroscience, I think that what we're seeing already from companies working on CRISPR and chimeric antigen T cell receptors and similar things, is that the bone marrow transplants generally and genetic engineering even more broadly are incredible tools to change our genes in specifically designed and engineered ways to fight specific diseases. And that can be diseases we inherit, diseases that we acquire, like cancer. And so just the ability to modify the way that our cells work, since our cells are such powerful tools, and especially in the case of the blood system, our immune cells, I think that's a huge role that stem cell biology is already playing in medicine. In the geroscience context, I think that we have a whole number of other stem cell approaches that are kind of coming into their own. And these are mostly organ-specific stem cells. So this means like bladder, colon, skin, blood also relevant here, even neural stem cells. And this idea of being able to grow and replace pieces or whole cloth, entire damaged organs, is such a powerful idea. And it seems like we're able to do it, especially for certain, let's call them simpler organs, like bladders, trachea, that have shown excellent results in the clinic so far. And initially, this is for people with like very severe conditions, not just for like people that are getting old and say like, oh, well, I've been using my trachea for 80 years, I should get a new trachea now. Instead, it's people who have severe damage in these things. But I think that this trend is going to be relevant extremely relevant for geroscience, but it's kind of a next wave sort of relevance. Because stem cell biology is incredibly challenging and tissue engineering type approaches are so tough that we're going to see them developed bit by bit the way that they've been making progress. And it's been amazing progress, moving quite fast, but quite fast still means many years, right? As we get it right and then perfect the technology and make it broadly safe and applicable to lots of people. And so I see geroscience as using these stem cell therapies after the healthcare system generally switches over to this idea of we should replace or treat diseases, or replace tissues or treat diseases before they start breaking down. And for that, we have to have very, very safe, routine, effective organ rejuvenation therapies. And whether that's replacing organs whole cloth with tissue engineering approaches, or there have been interesting ones that have been published quite recently relating to like the reprogramming of cells or the rejuvenation of stem cells inside the body, which, I mean, it's yet to be seen, but I also think that those therapies may have a role to play in this. That in my mind is like second wave geroscience approaches, but is among the first wave of things that seem to be ready to jump from lab bench to clinic now, that groups like Apollo are fascinated by in our investing world.
What are some of the barriers besides regulation and insurance structures not being a good fit for stem cell and reprogramming therapies?
Yeah, so I think that stem cell therapies are worth discussing slightly separately from a regulatory standpoint, because there is still a big regulatory hurdle that some but not all stem cell therapies have made, which is the fact that if you're a regulator, how do you deal with the fact that every single cell you take out of a person, grow in a lab, and put back into a person is really behaves like a different drug, right? It's not like you're manufacturing the same lot of pills all in a factory and then giving one pill to each person. You're taking their specific cells, growing them up and putting them back into that person, or taking an iPS cell line and putting it into that person. And so one of the big issues that the stem cell world is grappling with right now is the autologous versus allogeneic debate. Autologous meaning, how do we regulate stem cells that come from a person and are modified outside the body and go back into that person, which has specific regulatory challenges, versus the allogeneic approach, which is we have a line of cells that we can then grow up, is quite uniform, can be looked at and regulated like a drug, so it has less regulatory problems. But when you try to play a catch-all solution in cellular biology, and you try to put those cells into multiple people, then you have all sorts of scientific problems, like immune responses and so on and so on. And so there's this scientific and regulatory push-pull on how can we keep therapies safe and effective for patients in a way that we ensure the highest quality, and ensure that what we're doing actually works for that patient. And then on the second hand, how do we build something that is scientifically really, really going to have the effect that we want? So that jumps to the next problem, which is how do you measure efficacy in stem cell therapies? And I think that the field is tainted a little bit because a lot of early people that were doing human clinical trials in what they called the stem cell field then did this in a very much wrong way, where because of this autologous allogeneic push-pull, there were a lot of clinical trials designed without substantial enough scientific thresholds to show true efficacy, which, I mean, don't get me wrong, is an incredibly complicated question because you're modifying entire organ systems, not just individual molecular pathways, right? And so because of these very, very challenging clinical trials that were set up and then showed middling results, it kind of has polluted the water a bit for follow-on stem cell therapies. And so I think that the strategy piece of this, how do you design and execute something that will go after a much needed target in a way that can really help patients in a quantifiable way that makes sense to regulators, is not at all a straightforward problem. The only other piece I would mention as far as like major challenges in stem cell therapies is that compared to manufacturing aspirin, putting it in a pill and sending it out to pharmacies, the cost of stem cell therapies is astronomically high by comparison, right? And that should be obvious. It's so much work to do each individual patient, and so many things can go wrong when you're dealing with biology as opposed to chemistry, that the companies that are really trying to move this to a mega scale and industrialize the delivery of stem cell therapies have enormous cost challenges that they have to deal with, because they want to try to keep the cost of the therapies as low as possible while still making enough money to pay for the years and years of R&D that came before, and clinical trials and so on that came before launching the drug, and also to fund their work on the next more improved version and the next more improved version, while also giving some kind of financial return to their investors who took 20 years of patience and risk in developing that drug. And so that cost piece is a very big challenge. And we've seen some innovative therapies, especially on the gene therapy side, struggle with this already, where the initial price tags have been so high that it's caused some backlash.
Are these problems specific to tissue engineering approaches? Would reprogramming strategies be able to dodge them?
I think that with reprogramming, it depends on how you would approach it. It really depends on the therapeutic actor. So you get around some of the problems no matter what, because no matter, like, if you use a virus or a small molecule or a cocktail of molecules to do your reprogramming approach, lipid nanoparticles with a plasmid inside to modify the DNA, those sorts of things are all cheaper than trying to grow new organs whole cloth outside of the body for every patient. And so you end up with less cost problems there. But as we've seen from the gene therapy space, there are still major issues on the cost side, simply because doing this in the right way, and I think in the reprogramming world, you're definitely going to run into this, because there's a very serious concern for an expected side effect, which is the creation of teratomas within tissues. And so the bar for safety on a reprogramming therapy is going to have to be set so incredibly high that we'll have to just be very, very, very careful and thorough in assessing assessing any putative reprogramming agent as we move to clinical trials in humans.
So we've heard a lot about the funding and investing that's happening in the geroscience space. Why are investors taking an interest in this now?
Yeah, I think this is a really interesting topic for us, and obviously it's the reason that we exist, right? Apollo is a relatively new fund. We're about 2 years old, and we probably could not have existed 5 years ago. There's another group that we should mention if we're talking about investing in the space, Laura Deming from the Longevity Fund. She was around, I think was really a pioneer there, and did some early investments with a longevity focus. But overall, I would say that the momentum for aging investing has really ticked up in the last 2 to 3 years. And I think that the reason for this is somewhat simple to explain. And I call it the Geroscience sandbox. So let me explain what the Geroscience sandbox is. Since Cynthia Kenyon showed that we could extend the lifespan of a worm by modifying its insulin signaling pathways, the geroscience world has been in a kind of interesting state, because we've been able to say, first in worms and then also in flies, yeast, and then mice, hey, we've just done a thing that extends the lifespan of this organism by a substantial margin. And we didn't always used to mention that, oh, by the way, that means it gets less cancer and less Alzheimer's disease and less sarcopenia and less arthritis. We would just say it extends the lifespan of this organism. And can we do anything with that? And people until a couple of years ago would say, like, no, there's no regulatory path for that. You can't just go launch a drug for aging. That doesn't make any sense. Clinical trials take too long, et cetera. And so they would say, like, okay, well, I guess I'll just go back and figure out how I can modulate that pathway better to extend the lifespan of the animal even more. And so you had this kind of academic cycle where we were playing, as I call it, the sandbox. We were playing with all this really interesting biology and improving upon it. And it was just isolated from the rest of the world, that as cancer biology and dementia biology made these enormous charges since 2000 from, like, genetic screening, huge assays, tons of money pumped into them, and then rushing to the clinic. Meanwhile, all of these interesting parts of biology were kind of being played around in the geroscience space with much less focus from pharma and investment groups on like how can we turn that into something that can go into patients. Despite the fact that I would argue from a very early stage, it was obvious that the things that were going on here had enormous potential value to patients. And so I would point to some work that was done not solely by him, but I think exemplified by him, which is the work from Walter Longo, which over the past few years has shown that if you take the best known longevity enhancer, right, caloric restriction, or fasting, or a fasting mimicking diet, you can actually start using this in interesting ways with actual therapies today. So in addition to just fasting to prevent disease, Walter has all of this excellent data showing that a fast before chemotherapy sensitizes cancer cells to the chemotherapy and protects the non-cancer cells. And so this idea of using the aging-related biology to treat or become a part of medical treatment for existing diseases and not just being used as a general prophylactic for diseases, that I think has started to push a switch in people's minds. And again, to emphasize, it's not just Walter's data, it's a huge amount of research across all sorts of different mechanisms of action, but I think nicely exemplified there. And we've just seen this cascade of like, okay, yes, if you turn on autophagy, right, you can extend lifespan by driving an upregulation of autophagy. But also autophagy plays a role in all sorts of diseases like Parkinson's disease and Alzheimer's disease and even diabetes. And so each of these things, you can modulate these pathways that we found in some ways in the Geroscience space from aging biology, and then look at the broader picture and say, like, these have applications to real diseases. And on top of that, for me as an investor, when you lift this veil, you say like, oh, you guys have been working on this for 20 years and really know a lot about this biology that hasn't been shared with the rest of the world in the way that pathways involved in cancer have been. And so for that reason, as an investor, there's kind of this feeling of like, okay, the curtain has been thrown off and a curtain has been lifted, and there's all of this incredible biology and incredible science behind this this research on aging biology, and how can we use that? Now we have some strategic tools and business tools to use that didn't exist before, and I think that's been the shift.
So where do you see things going from here?
So this is the part where I put the little disclaimer that's like, you're about to experience forward-looking statements that cannot be guaranteed of whatever. So I would say that I tend to be optimistic, but I like to think of myself as a conservative optimist. And so with that framing, I would say that I think Geroscience space is going to change the way we do healthcare dramatically. I think that research on aging biology is going to be as fundamentally disruptive to human medicine as research on vaccines and antibiotics were. Because the 21st century is going to be defined by a switch in thinking about the diseases of aging as things that we have to wait to happen to people, diagnose, and then treat, to things that we know will happen in people and use approaches to prevent them. The same sort of way that we used vaccines to, instead of waiting for someone to get big swollen lymph nodes when they have bubonic plague, and this is more antibiotics, but whatever, or wait for someone to break out into smallpox, say like, okay, we're gonna treat your smallpox first, because we know once you get smallpox, it's very difficult to treat. Cancer and Alzheimer's are the same. We just haven't had the tools to intellectually approach the clinical treatment of cancer and Alzheimer's in that way until now. But I think that as the research improves, we're going to make that switch as a society to start thinking about cancer, Alzheimer's, late-stage diabetes, sarcopenia, osteoporosis, et cetera, as diseases that we can interfere with early in order to prevent them from happening instead of waiting for people to get sick. And so I think that as we create new medicines that are targeted at these specific conditions, as we hit regulatory milestones that broaden the applicability of these medicines to the prevention of human disease, there will at some point, and I think it will happen gradually, and it will happen over the next 10, 20 years, we will experience a shift from waiting for ourselves to get sick of the most deadly diseases in humanity to proactively using the power of modern medicine, not just the power of diet and exercise and supplements, but the power of modern medicine, molecular targeted therapeutics, gene therapies, stem cells, etc., to prevent those diseases. And for that reason, I think that this focus on geroscience is the biggest medical opportunity of the century.
When you say early prevention, you don't mean we start treating people at like 55.
It can be either. I think that the age limitations will move down and down and down and down as proof for efficacy goes up. It's too early to say. When I built economic models of what a geroprotector would look like, I usually actually start treating people at 55 in the models that I build, because mostly people who are 55 are not suffering from the diseases of aging, mostly. They've certainly accumulated damage, but if you have a battery of treatments that can both rejuvenate and prevent, then you can make a big difference, I think, at 55, or I think the TAME trial for metformin enrolls patients at 65. That's 65. But I think that this barrier comes earlier and earlier and earlier as we have more and more evidence to show that treating early helps.
It's also a question of safety, right?
Yeah, and so for all of these treatments, and this is something that should underline this whole conversation, is that as we make the transition, similar to vaccines, for example, as we make the transition from waiting for someone to get a disease and treating them to a preventative disease, the bar for safety becomes incredibly high. Because if there are even rare events, unless they're super rare events, but even rare clinical adverse events or side effects are not acceptable in a prophylactic. And so that means either knowing who will respond and taking them off the drug early, or preventing them from ever going onto it, or just building treatments that are exceptionally safe. And so I think we have to hold ourselves to just a really, really, really high bar there.
Well, James, thank you so much for talking to Geroscience. It's been a lovely and informative conversation.
Well, yeah, this has been super fun, Tegan. Thank you for all of your questions, and I hope your readers and, I guess, viewers enjoy it.